All right, welcome back. So this is part 2 of microbial control, physical agents. Okay, so we talked about sterilization and disinfection are processes. So that last part, disinfection, any process that destroys the vegetative pathogens. So I think I hurried up very quickly at the end of the last video, but essentially we are getting rid of all the vegetative cells. In disinfection, we cannot get rid of endospores. So that's the difference between sterilization and disinfection. So let's take a look at the agents used in these two processes. Now, when you see the word side, we're thinking that we're killing something. So you'll catch on pretty quickly. So bactericide, chemical that destroys bacteria, except for those in the endospore stage. Okay, so we're killing the vegetative cells of the bacteria. Fungicide is essentially a chemical that will kill fungal spores and hyphae, and there are specialized cells of some fungi in a particular group, and we call these the yeasts. So we're going to go ahead and put that there. So fungicides kill spores, hyphae, and yeasts. Viricide, clearly going to kill or inactivate viruses, and we're especially thinking about on living tissues. Sporicide, an agent capable of destroying bacterial endospores. And then lastly, just kind of a broad term, germicide, a microbicide, chemical agents that kill microorganisms. So when you see side, you're thinking to kill something, but as we talked about with the bactericide, it's only killing the vegetative cells of the bacteria, not the endospore stage, which again, we are going to set that as our highest level or our highest standard for being considered sterile is being completely removing microorganisms, including the endospore stage. Okay. All right. In living tissues, we're often thinking about these two terms, sepsis, asepsis, and then antisepsis. So you probably heard someone say, for example, my dad frequently had pneumonia due to aspiration of food and drinks, and essentially he had pneumonia and he went septic. So what that usually means is the infection is no longer contained to a particular organ or part of an organ, but it is spread into the blood. So when we have sepsis, we have bacteria in the blood, which creates a problem because it can multiply very rapidly. Of course, it could take up residence in other parts of the body, but essentially this is very serious. Asepsis then, A means without. Asepsis means essentially any practice that prevents the entry of infectious agents into sterile tissues and thus prevents infection. And then we have our, of course, aseptic techniques. So cleaning your hands, washing your hands or using hand sanitizers before you enter a patient's room. And when you leave that patient's room, if you are inserting a catheter, make sure you're doing that aseptically. So there is a particular process that you have to follow so you're not potentially contaminating any of the equipment that you might use to do that procedure, especially if it's an indwelling catheter. You wouldn't want to inadvertently contaminate that, causing essentially potential for a UTI infection. Okay, so what are antiseptics? We've all become very familiar with antiseptics in probably your entire lifetime. I remember a time when there was not antibacterial soap. It was just soap, like everything was soap. It wasn't even in a pump, it was just a bar. You picked up the bar, you washed your hands really well, and maybe you even went into the bathroom and there was like a towel, a literal cloth that was wrapped up in this machine, and you would pull that cloth and it give you a clean surface, and then essentially there would be laundry services that would collect those and then clean them and then make sure they were free of microorganisms, and then they would reinstall it in the machine. These days, we have air dryers and paper towels, and of course we have a wide variety of different kinds of soaps, but essentially we've gone back to soaps that don't have antibacterial properties or chemicals in them because we know that that can lead to antibiotic resistance. Okay, so what are aseptics or antiseptics? Antiseptics are chemical agents applied directly to exposed body surfaces. So think skin and mucous membranes, of course, wounds, surgical incisions, and we are trying to prevent the contamination by vegetative pathogens. All right. So I mentioned earlier, if you use alcohol swab, we're swabbing from the inside out on a patient's deltoid, and then maybe we give them a vaccine or we give them a shot, and that's going right there in that muscle. So we want to prepare that ahead of time. So as you're puncturing the skin with the needle, you aren't potentially taking skin bacteria and inserting it into now the wound that you're forming. Certainly, if you are doing your, let me get this right, you're doing your skills check off in the nursing skills lab, and so you're going to have to demonstrate that you can clean a wound appropriately. So there's going to be a method to do that, to ensure that you're doing it to reduce the number of microorganisms in that wound and also not introducing new microorganisms. We talked about using chlorhexidine on your arm when you give blood. Same sort of thing as putting a needle into your arm for an injection. You might remember during COVID that if you went to the dentist during that time, that the dentist would ask you to swish with some hydrogen peroxide. So essentially, it was a weak solution of hydrogen peroxide, but what it was going to do is drastically reduce essentially microorganisms that could be in your mouth, including COVID, so that the dentist could clean your teeth without potentially exposing themselves to any infections that you might have, particularly COVID. And then, of course, ordinary hand washing is says a germicidal soap. Oftentimes, it doesn't take any special chemicals in soap, and we will learn this in the chemical control of microorganisms. But essentially, washing your hands with soap is adding detergents to your hand, which is releasing the oils, and is also allowing you to eliminate dirt or maybe some other chemicals that are on your hands, like hand lotion, or if you were, I don't know, canning something and you had your hands in some vinegar or whatever. So essentially it's helping you remove those oils and debris, and then turns out that we're also washing away bacteria with it. So our normal everyday soaps are really great for reducing microorganisms on our hands. Okay, agents versus processes. So this is just the fourth slide, hence the little subscript there. All right, so when you see the word stasis or static, it means to stand still. But what we're going to think about it is bacteriostatic or fungistatic essentially means that we are inhibiting the growth of whatever it's referring to. So bacteriostatic, chemical agents that prevent the growth of bacteria on tissues or on objects in the environment. So it's reducing the growth to stand still, preventing it from overpopulating. Fungistatic, chemicals that inhibit fungal growth. Antiseptics and drugs or medications are often microbistatic because essentially because we are reducing the growth of the microorganisms. We may not get to a point where we've eliminated every single one of them. So I'm thinking about antibiotics, but you've drastically reduced it so that your own natural populations of bacteria in your body can outcompete whatever might be left. So when we think about a cidal agent, it does not mean sterilization. It just essentially means that we have dramatically reduced the growth. Okay. Don't need this slide. Okay. Okay. So kind of an interesting slide here, thinking about other considerations of microbial control. So substances. What substance are we trying to sterilize? Because then we need to think about how to not break down that particular substance or compromise it in any way, but make sure that we're eliminating all those vegetative pathogens, forms of the pathogen, as well as the endospores. So we got situations requiring sterilization confront persons involved in healthcare. So again, there are aseptic techniques or sterile procedures. but essentially we need to make sure that we're not compromising the skin or compromising, the joint or maybe another body part that's being operated on. So you see no universal methods work well in every case. So other considerations, how much does it cost? How effective is it? And then what is the method of disposal? So we might be able to sterilize, for example, in our microbiology lab, we can sterilize all of the petri dishes that we've used. And then what do we do after we put it in the autoclave for that usually is 20 minutes. So what do I do with those plates after that? Generally speaking, I can throw them in the trash at that point. But when we're talking about maybe all the medical waste out there in the hospital, what do we do with all that medical waste, which, has been contaminated, if it's, endotracheal tubes and so on, you can only use that once and then, we got to chuck it. But it could be actually covered with very pathogenic Pseudomonas, for example. So how are we going to get rid of that? What's the cost of that? I'm not sure why those slides were there. I think this may not be my final. Okay. Okay, so for those of you that are really into the biology of microbiology, this table gives you a really good idea how our physical and chemical agents are going to act on cells. Okay, so remember cell walls. When I think about cell wall properties of fungi, they are different than the cell walls of bacteria, which are different than the capsids of viruses, right? And when we talk about the envelopes of viruses are different than the lipopolysaccharide layer of our bacteria. And I know that fungi don't have that. So when we think about the properties of cell walls, we can target our agents to those properties. Okay, so it mentions you can block the synthesis of the cell wall, or maybe your chemical agent actually just digests it, which is kind of neat. So we have our chemicals, detergents, and alcohols over there. Okay, the cytoplasmic membrane. So thinking about membranes, we know that viruses don't have membranes like we do in our cellular or our living cells. So we can target agents to that lipid layer of the cytoplasmic or plasma membrane. And when we do that, we're essentially opening up the cell and disrupting its homeostasis. So detergents and alcohol could do that. Cellular synthesis. So this is pretty fascinating, and we're going to get more into this in next week lectures on antibiotics and antivirals. But essentially, we can target our agents, and I'm thinking in particular chemical control agents, directly to the synthesis of proteins via the ribosomes. So the ribosomes of prokaryotes are different than the ribosomes of eukaryotes, and we know that viruses don't have them. So they're going to hijack your cellular machinery and use your ribosomes or a bacteria's ribosomes. So essentially then we can target those proteins, so some part of protein synthesis, and our chemical agents are very good at that. And radiation, which is a physical control. So we got cellular synthesis and then a little bit more about proteins there at the bottom. Okay, I think I'm going to stop here and we will start a new lecture in a minute.